In a cohort of 9,000 adults spanning ages 18 to 101, researchers found that people whose gut microbiome became more compositionally unique, more distinct from everyone else's as they aged, were associated with lower serum levels of tryptophan-derived metabolites, higher levels of certain indole metabolites, and a 12% reduction in all-cause mortality risk compared to those whose microbiome became less unique, more average, over time. The strongest signal of healthy aging in this dataset was not a specific probiotic, not a perfect diet, but the degree to which the gut microbiome diverged from the population norm. In centenarians, that divergence was remarkable. Understanding what drives microbiome uniqueness and what destroys it is the subject of this video. For most of the history of microbiome research, the dominant assumption was straightforward: more diversity equals better health. A richer ecosystem should be more resilient, more species, more functional redundancy, more adaptability. This assumption is intuitive and partly correct. But the Wolmansky nature metabolism study introduced a more nuanced concept. It is not just richness that predicts survival, but distinctiveness. The microbiome that predicts the best outcomes is the one that is most itself, most differentiated from the population average, rather than simply the one with the most species. This reframing has significant practical implications, because it shifts the question from how do I add more species to my gut, to how do I support the ecological individuation that produces a uniquely healthy microbiome. The two questions have somewhat different answers. In this video, we will cover what the Wolmansky nature metabolism study found about microbiome uniqueness and survival across 9,000 adults, why the finding differs meaningfully from the more familiar concept of species diversity, what the blood metabolomics data reveals about the mechanisms linking microbiome composition to systemic health as we age, and what dietary and behavioral levers actually shift microbiome composition in adults over 55, including what the fermented food trial evidence specifically shows. Quick note before we begin: this is educational content based on published research, not medical advice. Talk to your physician before changing your regimen. Now, the study. The human gut microbiome is a community of trillions of microbial organisms, bacteria, archaea, fungi, and viruses inhabiting the gastrointestinal tract, with the highest density in the colon. The bacterial component is the most studied and arguably the most functionally consequential. These bacteria perform functions that human cells cannot, fermenting dietary fibers into short-chain fatty acids that feed the colonic lining and modulate inflammation, synthesizing certain vitamins including vitamin K and B vitamins, educating and calibrating the immune system through constant antigen presentation, maintaining the physical integrity of the gut barrier against pathogens, and sending metabolic signals to distant organs through the portal circulation and the gut-brain axis via the vagus nerve. The microbiome is highly individual. Two unrelated adults share only about 30 to 40% of their gut microbial species on average, far less similarity than in any other human body compartment. This individuality is shaped by early life factors including birth mode, breastfeeding, and antibiotic exposure, as well as by lifetime diet, geography, illness history, and ongoing antibiotic exposure across adulthood. For most of early adulthood, gut microbiome composition tends to be relatively stable within a given individual, while continuing to vary substantially across individuals. What happens to the microbiome in middle age and beyond was, until recently, considerably less well characterized, partly because microbiome research relied heavily on convenience samples of younger adults, and partly because large cohort studies with both longitudinal microbiome sequencing data and long-term health outcome data were rare. The dominant assumption in the microbiome field for many years was that higher alpha diversity, more distinct microbial species represented within a sample, was the key marker of microbiome health. This is ecologically intuitive. A richer ecosystem is generally more resilient and functional. And alpha diversity is genuinely associated with health outcomes in some contexts. But the Wolmansky nature metabolism study introduced a finding that added complexity to this framework. It was not just the richness of the microbiome that predicted survival, but its uniqueness relative to the population, how much it differed from the average microbiome pattern. An older adult with a highly distinctive, idiosyncratic microbiome, even if not the most species-rich one in the dataset, had meaningfully better survival outcomes than one whose microbiome had converged toward the population average. Ecological individuation, not just richness, was the signal. A study published in Nature Metabolism by Wolmansky and colleagues analyzed gut microbiome data from 9,000 individuals ranging in age from 18 to 101, drawn primarily from two independent cohorts, the Arivale cohort, a longitudinal wellness program based in the United States that collected repeated microbiome samples alongside comprehensive health data, and the Twins UK cohort from the United Kingdom, which provided an independent replication dataset with genetic and health data on thousands of twin pairs. Both cohorts provided gut microbiome data derived from 16S ribosomal RNA sequencing of stool samples, the standard methodology for characterizing bacterial community composition at the genus and species level, along with comprehensive health assessments, blood metabolomics panels, and mortality outcome records. The researchers defined microbiome uniqueness operationally as the inverse of compositional similarity to other individuals within the same age range, essentially a measure of how different an individual's microbiome was from the microbiome average of their age-matched peers. An individual whose microbiome closely resembled the average of their age peers scored low on uniqueness. An individual whose microbiome was compositionally distinct from peers scored high. This metric was tracked longitudinally in participants with repeated microbiome sampling to assess whether microbiome uniqueness was increasing, decreasing, or stable as individuals aged over the follow-up period. The analysis then linked these uniqueness scores and trajectories to two important outcome dimensions: survival data from mortality records, and blood metabolomics data measuring the circulating levels of specific metabolites that the gut microbiome produces or strongly influences. This metabolomics layer was a key methodological contribution that distinguished this study from earlier microbiome aging research. Rather than stopping at the bacterial composition level and leaving the mechanism implicit, Wolmansky and colleagues traced the specific biochemical signals that compositionally divergent microbiomes were producing in the systemic circulation, providing a plausible biological mechanism for how microbiome composition could plausibly influence health outcomes at a distance from the gut. The study adjusted for age, sex, body mass index, and other demographic confounders, and conducted multiple sensitivity analyses to ensure the uniqueness survival relationship was not driven by specific confounders including dietary patterns, medication use, or disease status at baseline. Critically, the findings were replicated independently in both the Arivale and Twins UK cohorts, a methodological strength that substantially increases confidence in the generalizability of the association. The researchers found that microbiome uniqueness increased with age in the healthy, long-lived participants of the cohort, particularly among those who survived to advanced age and maintained good health status, and decreased or remained stagnant in those with poorer health trajectories. Individuals whose microbiome uniqueness was in the higher quartiles of the distribution showed a 12% lower all-cause mortality risk across the follow-up period compared to those in the lower uniqueness quartiles. This is a meaningful effect size for a single microbiome-derived metric. In centenarians, adults who reached 100 years of age or above, the uniqueness signal was particularly pronounced and striking. The gut microbiomes of centenarians in this dataset showed compositional patterns that were genuinely different from those of adults in their 60s and 70s, not just incrementally more diverse, but distinctly different in the dominant bacterial taxa present and in the metabolite profiles they produced. This observation is consistent with findings from independent studies of centenarian populations in Japan, Italy, and China, which have noted distinctive gut microbiome patterns in exceptionally long-lived individuals compared to their less long-lived peers. What the researchers found next is even more important for this age group. The metabolomics data provided a mechanistic window into why microbiome uniqueness might matter biologically. Individuals with high microbiome uniqueness had lower circulating levels of tryptophan-derived metabolites processed through the kynurenine pathway, a metabolic route associated with inflammation, immune dysfunction, and neurodegeneration, and higher levels of indole-derived metabolites produced when specific bacterial species process tryptophan through an alternative pathway. Indoles are increasingly recognized in the microbiome literature as anti-inflammatory signaling molecules that influence immune regulation, gut barrier integrity, intestinal motility, and potentially brain health through the gut-brain axis. This metabolite pattern provides a biologically coherent mechanistic story. A compositionally unique microbiome may produce a distinctive circulating metabolite fingerprint, with relatively higher levels of protective indole derivatives and lower levels of pro-inflammatory kynurenines. These systemic signals may then contribute to reduced chronic inflammation, better immune regulation, and ultimately better survival outcomes. This is a correlation, not a proven causal chain, but it is mechanistically plausible in a way that makes the uniqueness survival association considerably more interesting than a simple statistical association. The limitations of this evidence deserve careful attention. This is an observational study, and the core limitation of observational microbiome research is the difficulty of establishing causality. The finding that microbiome uniqueness predicts survival does not prove that changes in microbiome uniqueness cause improved survival. A genuine alternative explanation exists: the same underlying genetic factors, lifestyle factors, and early life environmental exposures that produce good longevity outcomes may also happen to produce more distinctive microbiomes, meaning the microbiome is a marker of underlying health and longevity capacity rather than a driver of it. Separating the marker from the cause requires intervention studies that directly manipulate microbiome composition and measure survival outcomes, experiments that are logistically difficult, ethically complex, and have not yet been conducted in humans at the scale necessary to answer this question. Animal experiments using germ-free mice colonized with microbiomes from young versus old donors, or from long-lived versus average lifespan animals, provide mechanistic support for the direction of causality, but the translation from germ-free mouse microbiome experiments to human aging biology involves substantial inferential leaps that should be acknowledged honestly. The measurement of microbiome composition from stool samples also introduces technical variability. The microbiome is not homogeneous across the gastrointestinal tract. The composition of the distal colon, which stool samples reflect, differs from that of the small intestine and proximal colon. Stool sampling misses potentially important microbial populations in other gut regions. Sequencing depth, primer choice, and bioinformatic pipeline differences across studies make quantitative cross-cohort comparisons imperfect. The practical question that most adults will ask, what can be done to shift a microbiome toward more uniqueness, is not directly answered by the Wolmansky study, because uniqueness is an emergent ecological property of a complex community rather than a simple trait that can be targeted with a single intervention. What the broader dietary microbiome literature most consistently supports, and what the takeaways in this video draw on, is that dietary fiber variety, fermented food consumption, and protection from unnecessary antibiotic disruption are the most evidence-grounded modifiable levers for supporting microbiome health in adults. 3 evidence-grounded takeaways for adults over 55 engaging with the microbiome literature. Takeaway 1: dietary fiber is the most consistently supported lever for microbiome health. Fiber is the primary substrate gut bacteria ferment to produce short-chain fatty acids, butyrate, propionate, and acetate, which fuel the colonic lining and have systemic anti-inflammatory effects. Target 25 to 38 grams per day from whole food sources: vegetables, legumes, whole grains, fruits, and nuts. Variety matters. Different bacterial species ferment different fiber types, so rotating through different vegetables and legumes supports compositional diversity better than eating the same foods repeatedly. Takeaway 2: fermented foods show distinct evidence for modulating the microbiome in ways fiber alone does not replicate. A randomized trial by Waystick and colleagues from Stanford, published in Cell in 2021, found that a high-fermented food diet for 10 weeks increased microbiome diversity and decreased 19 distinct inflammatory proteins, an effect not seen in the high-fiber group. Yogurt, kefir, kimchi, sauerkraut, miso, tempeh, and kombucha are traditional foods with centuries of consumption history. The evidence for their microbiome role is more robust than for most commercially sold probiotic supplements in healthy adults. Takeaway 3: protect the microbiome from unnecessary antibiotic exposure while never avoiding antibiotics when genuinely medically necessary. Antibiotic courses become more common after 55, dental procedures, respiratory infections, urinary tract infections, post-surgical prophylaxis. The microbiome evidence shows that antibiotic courses produce substantial disruption of gut communities with recovery timelines measured in months. The practical approach is informed conversation with a prescribing physician, asking whether a broad-spectrum agent is necessary when a narrower spectrum one would suffice, or whether the condition requires antibiotics at all when this is genuinely ambiguous. This is not antibiotic avoidance, it is informed stewardship of a microbiome asset the Wolmansky data suggests has meaningful longevity implications. The Wilmansky Nature Metabolism study adds an important and nuanced dimension to how we think about aging and the gut microbiome. It is not just about having a diverse ecosystem, it is about maintaining a distinctive one. The survival implications of that distinctiveness, observed consistently across 9,000 adults including centenarians, with blood metabolomics data providing a mechanistic window into how microbiome composition connects to systemic health, make this study a significant contribution to longevity science. The most practical and evidence-supported levers remain dietary fiber variety from whole food sources, regular fermented food consumption, and thoughtful stewardship of antibiotic exposure. None of these require expensive supplements, exotic interventions, or commercial probiotic products with limited evidence in healthy adults. The PDF guide on reading your biomarker panel, linked in the description, includes the inflammatory and metabolic markers most relevant to the biological pathways connecting the gut microbiome to systemic health. Subscribe to stay current with this rapidly evolving field. This is educational content, not medical advice. Always consult with your physician before changing your regimen. Studies are preliminary, individual responses vary.